welcome to the Current Science and Technology podcast from the Museum of Science in Boston. I'm your host, Corrine Tate, and every week we bring you interviews with guest researchers and our museum staff covering science and technology in depth. My guest today is Jessica Winter, a professor of chemical and biomedical engineering at The Ohio State University, who is also an inventor and entrepreneur in the midst of commercializing one of her nanotech inventions. Welcome, and thanks for joining us for this phone interview, Dr. Winter.
Well, thank you so much for inviting me.
So we know you already through our work together on the QStorm Collaborative Research Project. And for that project, you're engineering quantum dots that could be used to produce incredible super resolution microscopy images of tiny processes that are occurring inside living cells. And our listeners can find out more about that project at qstorm.org. But today we wanted to talk to you about a new company that you formed to do something else with quantum dots. Can you tell us a little bit about that?
Sure. So we founded Core Quantum Technologies in 2012. The main focus of Core Quantum is to develop imaging agents for pathology. So when you go see a doctor, you often have a blood sample taken, and occasionally you might have a tissue sample taken, for example, a biopsy. And that sample then gets sent to a lab where it's analyzed to look for a specific disease or pathology state. And that analysis includes elements that label certain parts of a cell and light them up so the pathologist can see them. Right now, those elements are chemicals, and we are developing a new type of nanoparticle, a quantum dot, that can replace those chemicals. There's basically two main advantages that this technology would offer. The first is that it can be brighter than what's currently used. And brighter for a patient means better sensitivity. We're more likely to detect that cancer or that blood disease. So that's important to patient diagnostics. The second benefit is that we can make them in lots of different colors, which means that we can label many different parts of the cell all at the same time. And that allows the physician to look at how different chemicals or different proteins and elements within the cell are interacting. So right now, people usually label one thing at a time on separate slides, and that doesn't tell you about the interaction.
In what kind of medical test would that interaction be important to identify or detect?
So our primary market right now is looking at cancer diagnostics. And in cancer, there are already several markers that have been identified for specific cancers as important. So for example, in breast cancer, estrogen receptor, progesterone receptor, and the HER2 marker are used to diagnose patients. So that would be sort of the initial. But as people develop drugs that target those markers, they want to look at what part of the protein cascade is the drug acting on. And this type of test would allow you to see that, and then that can help your doctor design a better chemotherapy cocktail for your treatment.
This seems to be a better test, not just because it can do everything all at once, but it could actually help inform your doctor on designing a better treatment?
Right. So that's a longer-term goal. Initially, we just want to replace the current reagents that are being used based on the brightness and sensitivity. But the longer-term vision is that this type of agent will enable physicians to develop more personalized medicine approaches where the treatment is tailored to each patient based on their specific expression pattern of genes and proteins.
So first off, we're making kind of a better, faster test. But ultimately, there's a lot more applications where this could take us that would really help patients.
Right.
Now, how is it that you picked this type of problem to focus on?
I actually started my research working on quantum dots, which are these materials that we're talking about. They're basically nanoparticles made of semiconductors, the same thing that your chip is made out of in your computer. One of the properties that got researchers really excited about them is that they produce light when hit by light of another wavelength. And so that allows them to be seen. So I was very interested in biomedical imaging, and I have been for some time. And so that's an obvious place for quantum dots to find application.
So I also heard that you created a magnetic version of quantum dots. What would those kinds of things be used for?
So magnetic quantum dots can be used for a couple different things. One application is still within the imaging realm. We do already have medical imaging based on magnetics, which is MRI, magnetic resonance imaging. And so magnetic quantum dots could provide what's called multimodal imaging, meaning that you're getting imaging signals from two different types of wavelengths of energy, and that allows you to see something with better specificity, because if it doesn't have both signals, it's not real. So you could do a magnetic signal and a fluorescent signal, and where you find both signals, that's where the label is. The other application, which I'm really excited about, though, is because these materials are magnetic, that means we can manipulate them with magnetic fields. And we've already demonstrated that we can use these materials to isolate cells and even biomolecules from solution at a very, very small concentration. So we can isolate single cells, for example. So if you could imagine getting a pinprick of blood, putting it on a chip, and getting some kind of diagnostic information out of that, that could really reduce patient trauma in terms of comparing that to, say, a 5 to 10 ml blood sample. So we're really excited about the idea of combining the magnetic aspect to the quantum dots, so that now we have both imaging, visualization, quantification, and also a magnetic component, which allows us to separate things from a mixed solution.
Oh, that's interesting, using the magnetic component to separate it. These nanoparticles that you've invented seem to have so many applications. How did you figure out what was most promising in terms of commercializing it, creating a company to get these out there?
That's a good question. So I have to say, in the beginning, we didn't. We didn't do a very good job of that, because as a researcher, I saw the applications that would appeal to me as a researcher, and so I wanted to sell into the biomedical research market to other professors like me. But fortunately, I was selected, along with my business partner and a co-founder of the company, to participate in the National Science Foundation's I-Corps program. So I-Corps stands for Innovation Corps, and it's a program that's designed to help people like myself who are inventors but not yet entrepreneurs to learn about the commercialization process, so that we can identify technologies that are viable. And through that process, we interviewed 87 customers, potential customers, including pathologists and biomedical researchers and biologists, people that are experts in imaging and diagnostic tests. And through those interviews, we determined that the best market, the one that had the strongest need with the greatest market potential, was in clinical pathology. And that's why we decided to focus on that market.
Oh, that's interesting. So doing this research really changed the direction that you guys had originally set out for Core Quantum.
Correct. So originally the company was planning to sell to a handful of researchers who would be interested in quantum dots as tracking elements in their own research. And through I-Corps, we realized that that handful of researchers might represent as few as 10 to 20 people total in the whole world. So obviously that's not really a market. Whereas clinical pathologists that we met with were very excited about our product, and that market represents 500 million just for the fluorescent reagents that we would be competing with.
So now that you've started this company, you figured out who your customers are and how your product can best be used. How is your R&D going? Are you up and running with, manufacturing these nanoparticles?
To some extent. So I say that with caution, because given that this is a clinical product, we will require FDA approval before we can be sold for patient use. So we are in the process of navigating that FDA approval process. That said, we did our first patient test in January of 2013, so about a year and a half ago, and it was successful, so we were very excited, and this is basically just a proof of concept that says that our product does indeed work on actual patient samples. So from that point, we began developing the product in more detail. We did a smaller patient trial very recently where we're comparing our product against the current gold standard. And so we're working right now very closely with beta testers to try and develop the product into the optimal formulation for their use. And there were a lot of things that came up that we didn't expect from that. So the way that the product is typically sold to a researcher like myself is it's a vial of particles that aren't necessarily modified with any targeting agents. But the pathologist wants what they call a plug-and-play kit, where it says take 10 microliters of the yellow vial and put it on the slide for 5 minutes. So part of our product development includes developing procedures and product inserts to explain to people how to use the product in a consistent way so you get reliable results each time.
It's not just about having your product be this brighter, better particle. It also needs to be really easy to use.
Right.
My understanding is that these nanoparticles are incredibly small. First of all, how big are they, and how on earth do you manufacture something that tiny?
In general, the accepted definition is something less than 100 nanometers in size. They're smaller than a cell. You can pack 1,000 to 1,000,000 nanoparticles within a single human cell. So really small. The nanoparticles that we work with are about 30 nanometers in diameter. Again, is incredibly small. And one of the problems that happens when you work with a material at that size is you can't touch it. And it's often very difficult to see it as well. Even on a standard light microscope, you cannot visualize something of that size. I don't know what people think of when they think of nanomanufacturing. I sort of envision like a fancy Honda, but that's not really what it is. It's very different, and the challenges are completely different from traditional manufacturing. And that's part of the reason why it's taking some time for nanotechnology to enter the consumer product market and become available. There are basically a couple of routes that people use to make nanoparticles generally. One is to take something that's bigger and grind it down so you get a nano size. Another is to pattern it lithographically in the same way that you would make a computer chip. But the method that we use is chemical synthesis. So we start with basically individual atoms and we assemble them into this nanoparticle using chemistry and self-assembly techniques.
You're able to make these small batches, but that's not as many as your customers would need, for example.
Right. So the method that we developed in the lab to make these particles works really well. But in order to do a single mouse test, we would need 100 of them. So I can hire someone to sit around making batches of these particles 24/7, but it's not very practical in terms of commercial scale. So one of the initial challenges we faced was how do we make more? And our initial attempts, we just said we're going to do the same process but at bigger scale. And we did that, and even scaling it up 15 times, which is still not enough for a single mouse, it didn't work. So we had to step back and think about what we wanted to make and how we might be able to make it at a larger scale. And one of the things that we recognized is one of the main innovations that's revolutionized bulk manufacturing at the large scale, is the assembly line and the ability to make things in a continuous way. So we looked for a process that would enable us to make particles continuously. And I was fortunate to work with a collaborator in my department, Barbara Luz-Lussel, who uses a spray technology to make micro-sized particles. And she and I discussed and worked together and developed a new technology which allows us to adapt that technology to the nanoscale so that we can now continuously spray nanoparticles, and we can make them in bulk at many times the volume that we were making at the batch level.
So that technique, this spraying them continuously, that's what's going to allow you to make the quantities that you need to make your company viable.
Correct. And it also greatly reduces the price of manufacturing the particles, which enables us to create a product that can be priced comparably with what's currently being used.
Now, if we're talking about spraying nanoparticles and quantum dots, which I understand can be toxic, you are probably concerned about safety.
Yeah, so initially the process that we're using sprays the material through air and it's collected in a water dish downstream. So there is a concern for the health of the manufacturer, and in order to protect them, the instrumentation is enclosed in a chemical fume hood which is vented externally so that the fumes should remain within the enclosed space. One thing that comes up a lot when we talk to investors and other people interested in nano manufacturing is what are your capital equipment costs? And I keep having to explain that nano is very small. So my capital equipment, we're not like a refinery that you might find down in Houston or, a manufacturing facility like Honda. Our capital equipment can fit on a desk. It's very small because nanomaterials are very small. So it's easy to enclose them in containment devices. And so something we've started working on more recently is, can we spray directly into water so that we could have an entirely enclosed system where the product is basically being collected in a water flow, and the entire thing could be enclosed so it would minimize exposure for the manufacturer.
I thought you might be able to mention some of the ups and downs of starting a high-tech company like this and being an entrepreneur at the same time as you're carrying on your full research and teaching load.
Well, it is a lot more work than I thought it was, to be honest. And I can say that as someone with little experience in business before this venture, I think that I have probably made every possible mistake with this company that there is to make. But that said, we're still standing and we're still going strong. So this has been a tremendous learning experience for me. A lot of people that I've spoken with compare entrepreneurship to childbirth. It's painful and it takes a long time, but at the end you have this beautiful baby. And I think that that is probably an apt analogy. It really is a lot of work, a lot more work than I thought. There are so many different aspects that you have to understand, everything from marketing and sales to financials to legal to patent protection. So there's a lot of aspects that you need to cover. And, of course, having a great support system is crucially important. And we're very fortunate that the state of Ohio provides a lot of those resources. And we've taken advantage of many of them. But it is quite a job to do all of that, I acknowledge.
It certainly sounds like it.
So for me, you could have started this company several years earlier, but I didn't because I was focused on establishing tenure at the university, and I waited until that took place to start this. So now I can focus a little more on the business, and I've closely integrated my business and research as well. But you always have to save that time to go to the school play or to the violin recital to be there for your kids, and it's quite a stretch. So I think it's very important to remember to always take care of yourself and your family, but also to remember that you can have everything, but not all at once.
Those are really good words of advice. So thank you so much, Dr. Winter, for taking the time to chat with us today and share your story of commercializing this nanotech invention.
Well, thank you for having me.
That's it for this week's show, but be sure to come back next time for more of the latest in science and technology. This podcast is a production of Current Science and Technology at the Museum of Science in Boston, part of the Boston community for over 175 years. For more information, visit our website at www.mos.org/podcasts or email us at podcast@mos.org. Thanks for listening.